Hello, I'm Emily Leproust. I'm the CEO of Twist Bioscience. And today I'm very excited to talk to you about how we use our platform that writes DNA to antibody discovery. It's a very powerful thing, and this is done under our Twist Biopharma brand. So to start, I'll give you a very brief history of DNA synthesis. I won't go through all of it, but in 1955 was the first time that we were able to add two T's together. So just two base pair synthesis. In 1981, there was a very foundational paper from Marv Carruthers on how to use phosphamidite and beads to do DNA synthesis, and that's the same technology that we still use today. And then there's been massive development around engineering of platform, but now at Twist, we have a silicon-based platform that writes DNA, and we are literally printing billions of bases every year that we ship to thousands of customers. And so at Twist, on our silicon chip, we can make 1 million oligos at the same time. And that is in contrast to the 96 well plate, where our competitors can synthesize 96 oligos at the same time, so 10,000 times more. And so that is the differentiator that Twist Biopharma uses. From those oligos, they can build superior libraries, and we'll talk about that in the next few slides. And then they also have access to a limited amount of genes that are very important for further characterize and optimize antibodies. So how to work with Twist and Twist Biopharma? Well, we participate on the entire spectrum from discovery of antibody to optimization to clinical development all the way to IND. We are usually extremely good on the discovery and optimization side. And so there, you can partner with Twist. You give us a target, and we'll give you an antibody that's ready for preclinical development. Or at Twist, we also have developed our own antibodies. And so we have a pipeline of Twist discovered antibodies that are ready for licensing or spinning out. Let's first talk about what's a library, what's an antibody library, and how an antibody library is used for drug discovery. And so first things first, what is a library? Library, in the context of biopharma, is a tube where we have different molecules. So we have different antibodies, different mutants, and often we use phage display, but the idea is in the same tube, we can have millions or even billions of different mutants, and then we'll work to extract the right antibody from that library of billions of mutants. So next, let's look at what's an antibody looking like. So an antibody is a complex protein sequence, and that's what our human body creates when there is a pathogen that is introduced. And at the end of the antibodies, there are some hyper-viable regions. They are called CDRs, so complementary determining regions. So those are small regions that are hyper-viable. And when they come together, the human body creates many different versions of them, actually, again, billions of different antibodies. And then the right antibody that actually is able to neutralize the pathogen then gets amplified. And when you sequence those viable regions, you sequence those CDRs, they are not random. Here you can see the results of sequencing, and they have some very specific ratios of amino acid. There are some very specific rules about what is allowed and what is not. And so that is what we're trying to recapitulate with our platform. Is we're resynthesizing CDRs that are allowed or that have rules that are allowed in the human immune system. another look at the antibody, and the CDR regions are made of 60 unique residues, so 60 amino acids. So it's not a lot. It's very few. However, if you calculate what is the theoretical diversity of those 60 amino acids, and that is 20 to the power 60, 20 because there are 20 amino acids. And so that means that if you were to synthesize all the different combinations, there could be 10 to the power 78 combination. And that number is astronomical. It is just no way that anybody, even nature, cannot sample that much diversity. And so therefore you have to subsample. And you could make a library that has about a trillion mutants. And so what that means is that you could only screen 9 amino acids in order to have a library that can be made. And 9 amino acid is not enough. You really want to sample more. You want to sample all of those 6s. And so something needs to be done to contract the space that is surveyed and screened. And so there's a few technologies to do that. We'll go through 2 plus the twist approach. So one approach is to do what's called an NNK. And so you start from, you actually will synthesize the DNA part of the CDRs. Because you encode for amino acid comes from 3 bases, you will synthesize NNK. So the first could be ACGT, the second could be ACGT, and the third is a subsample, only 2 bases. And so when you do that NNK synthesis, you can create 32 different codons. And so you are making, you're sampling quite a bit of space, but that is just for one position, and you will have to do that multiple times. So the good news is that you can make a lot of mutants. The bad news is that you still have stop codons. The bad news is that you cannot control the ratios of amino acid, and you still have sequence liabilities that will be a problem downstream. So NNKs are very inexpensive libraries to make, but they actually are not really good, and they don't follow the rules of the human repertoire. So a slight improvement is a trim synthesis. So in trim, you synthesize all 3 bases at the same time. And so that is a little bit better. And now with just 20 codons, you're able to synthesize any amino acid. However, you can't choose your codon, so that's a limitation. And you still have stop. You can eliminate stop codons, so that's good, but you still have sequence liability. And then what's quite an issue, you cannot write full CDR sequence. So what that means is you cannot make mutants that follow the rules of the human repertoire, and that's a big issue. So fortunately at Twist, we have the silicon chip, and so what that means is that in the computer, we can choose all the mutants that we want, and then we can make them from scratch on the silicon chip, and then assemble them together. And so now we can represent all 20 amino acids, all codons, no subcodons. We can remove the liabilities, we can control the amino acid ratios, and we can follow the rules of the human repertoire. And so overall, when you compare the NNK approach to the trim approach to the twist approach, we have checks all along, and the huge advantage is this ability to only encode sequences that are in the human repertoire and to put multiple CDRs together. So that's the library that we have. And so we can make libraries from scratch. And so you can come to us exactly with your library design. We can make it. Or we have on the shelf some pre-made libraries. And I won't go through them, but we have libraries that are general purposes. We have libraries that are specifics for difficult targets, either GPCR libraries or ion channels or carbohydrate and so on. And so, again, when you come to us, we put to work all of those libraries at the same time. And so we have access to more mutant than anybody else, but again, they fully represent the human repertoire. So a quick example of what it looks like. I mentioned that in the antibodies on each chain, you have CDRs, you have 3 of them, the hyperviable region. So this is one example of one library. For one chain where in the hyperviable loop 1, the CDR1, we have 1,200 unique mutants. And CDR2, we have 1,600. And then CDR3, we have 2.5 million mutants. So you can see why you need a twist technology, because if you make oligos, 96 oligos at the same time, it would be almost impossible to make 2.5 million oligos, whereas for us, 2.5 million oligos is just 2.5 chips. And then we can put them all together. And so in this library, there's a theoretical diversity of 4 trillion mutants. And when you do a phage display experiment, we'll talk about it in the next step. Actually, there's a subsampling phage display. Typically, it looks at about 1 billion mutants. So even though in that library you have 4 trillion mutants, only 1 billion will be tested at the same time. So now that we've made the libraries, let's talk about the next step, which is how do we pick the winner for the library from the libraries? And so there we use phage display. And so what that means is that in the library, we synthesize the DNA, we create a library, and then that DNA, those billions of mutants, are introduced into bacteria. And then we introduce phage, and phage will soak in the DNA from the bacteria. And so effectively, whatever DNA you've made, that ends up into the phage. And the phage have a very particular capability, which is the DNA is inside the phage, and the phage will synthesize the protein that is encoded into the DNA on the surface. And so you see outside there's a little dots. Those are the protein that has been synthesized by the phage, and we can link the protein to the DNA because they're on the same phage. And then we can use some kind of binding experiment to filter out the phage that don't bind. Those are the ones you don't want, from the ones that bind. And the ones that bind, you can then remove them, analyze them. And because the DNA is still available, you can go back and reintroduce that to the next step. And so what that means is that at the first stage, you start with 1 billion mutant, but as you go from panning, as you go through the cycle here, you enrich for the better and better sequences. And at the end, you may get only about 1,000, the best of the best 1,000 antibodies from the starting libraries. And so again, you start from 1 billion mutant in a tube, you go through the process, and at the end you get the 1,000 best antibody. Then you reformat them, and so that's where DNA synthesis is useful from Twist, because from scratch we're going to reformat those IgG, and then we can characterize them with affinity testing and functional testing. And so if you see in 3 weeks plus 4 weeks plus 1 week, so in 8 weeks, you can go from an idea to hits. And we use the power of NGS sequencing to analyze the DNA, to analyze the antibodies that are being enriched. So at each round, there's some sequencing that happens. And then what that means is that then we can also use machine learning and artificial intelligence, AI, to help us cluster the different antibodies to pick the right ones to reformat. And so what that means is when you get 1,000 antibodies, some of them may be highly related to each other. And so you don't want to only study the highly related one, but if you can cluster out the different families, you can make sure that you sample each family and you get better diversity of antibodies. And then the next step is to do the functional testing and the affinity testing. And so there's a lot of automated instrumentation that we've put in place to produce the IgG, scale them up, again, functional test them, affinity test them, make sure that there is no endotoxin, and make sure they have the right purity. So that is all of the work that we do inside Twist. So now, at this point, we have a hit. We have an antibody that is pretty good. And so the next step is to do optimization. And so there we do the same thing. We start from an input, and then we use the software to create more mutants. So we, from the few winners, we recreate billions of mutants that are again fully human derived. Then we synthesize them, go through the panning, and after a second round, you end up with a fully human and optimized lead. In addition to that, we now have the Abveris mouse capabilities at Twist. And so Abveris, they use a immunization approach where instead of using a library, what they do is they take the target, they immunize a mouse with it. The mouse immune response creates more mutant. And then you, when you extract that antibody mix, that's your library. And you try to find the best antibody from that library. And so they have two advantages at Abveris. One is they have a hyper-immune mouse. And so what that means is that with the mouse of Abveris, there's a stronger immune response. And so what that means is that you get response for more epitopes. And as an example here, if you have a wild type mouse, a normal mouse, sometimes the region in red, they are the same between human and mouse. And so you'll never get an antibody against the red regions because it's just the same as human. However, if you have a mouse that has hyperimmune immune system, you will get more antibodies, and you will get epitopes. You will get antibodies against the regions that are similar between human and mouse. So you get more antibody. That's the first thing they have, is they have a hyperimmune mouse. And then the second thing they have is that they are a power user of the beacon technology. And so what that means is that using the silicon chips of the beacon, they're able to individually assay the binding efficiency of B cells. And then once they found the winner, the ones that bind the best, the ones that have the best functionality, they can extract them, sequence them, and figure out the winners that can then be reformatted with Twist. So overall, they have a better mouse, and because they have a power reserve of the beacon, they can more quickly than anybody else hone in onto the winner. So what that means is that at Twist, we now have all three methods of antibody discovery. One is the synthetic library we'll talk about first. The second is the Abveris approach using animal. And then last, we also, through partnership, use AI and deep learning. And so when you come to Twist with a target, we can give you a better discovery, better screening, and ultimately better candidates. So now, how can we use that to treat disease? So just a few examples of what we've done at Twist. One example is during COVID. When COVID happened, very quickly, it was defined or discovered that the COVID virus had an S protein on its surface. And so at Twist, we discovered antibody to the S antibodies that seem to be highly neutralizing against all the different variants. And so those antibodies are now spun out under a company called Revela. Quite importantly, the Twist antibody that now Revelar is pushing into the clinic, they are universal against alpha, beta, delta, and Omicron so far. And then we also found different antibodies that were effective against the ACE2 human receptor of human cell. Another bunch of examples of what we do is shown here, where those are actual projects we are running for our customers. You can see that we go after many different indications, so endocrine and metabolic disorders, cancers, autoimmune and inflammatory diseases, infectious disease, dermatology, neurological disorder, and we even now have programs for cat and dog therapeutics, because same thing, we can sequence human and learn the human repertoire, but also we can sequence cats and dog and learn the dog repertoire, the cat repertoire, and make libraries that are specific to them. So very broad disease indications. And then the modalities are also quite broad. We can, of course, make antibodies, but we can make VHS, which are smaller. We can make bispecific. We can even make TCRs. We can do engineered enzymes. So there's really many different ways we can go after, and all of those programs are being done with our customers. So with that, I'll make a quick conclusion. Again, in summary, we have the ability to make massive oligosynthesis, which we turn into making amazing libraries that follow the rules of the human repertoire. Then we can screen those libraries through panning using phage display, get in just a few weeks to lead that we can optimize. And we've done that now with dozens of partners. We have more than 60 programs completed where those antibodies are back with the customers to go to IND, and we can really accelerate drug discovery and go after targets that were very hard. So one of our specialty and positioning has been drug discoverer of last resort, and that is resonating very well with our customers. And so with that, I'll pause, and thank you very much for your attention.